So, good afternoon and welcome everyone. It's really great to see so many of you joining us today for this second session of the Daylight Awareness Week. This public event started yesterday. It was on the day of the equinox, when day and night are of equal length or almost equal. And this day also means for us, at least for us in the Northern Hemisphere, that we are entering this period where days are getting shorter and where we are getting less and less daylight. So we all know this feeling of low energy and sometimes even negative mood in winter. But what does the lack of daylight really mean for our health, for our well-being? How exactly is it affecting us? This is what we will discuss in a few moments, together with a really great specialist that we have the privilege to welcome today. And this question of the lack of daylight is perhaps particularly relevant in these times of lockdown that we have experienced, or that we are maybe still experiencing. But it is actually always a very important topic, given that our modern lifestyle makes us spend most of our time indoors and away from natural light. Before we start, I would like to just explain very quickly what is the Daylight Academy, which is the organization behind this event. And I will just share my screen, just a second. So the Daylight Academy is an international membership organization bringing together scientists from very different disciplines, architects, and actually all kinds of professionals working with daylight-related topics. What we mainly do is to organize different conferences, different kinds of meetings, where all these specialists can meet, can exchange their ideas, and be inspired. Through this interdisciplinary approach, we would like to foster innovation and also generate new and creative research ideas. Daylight is actually something that we experience every day, more or less. So often it is also something that we don't really think about. So it's just there, it's normal. But actually, we really should think about, because as I mentioned before, it has a huge impact on us and actually in all life on Earth. And that's why another important goal of the Academy is to disseminate the knowledge. So all this knowledge that we have from our experts, we would like to communicate to the public and so create more awareness about these questions. And that's what we are doing today and what we would like to do with this whole Daylight Awareness Week. I also would like to emphasize that you as participants are really an active part of this webinar. So we would like it to be interactive, and you will have different possibilities to get involved. And I especially encourage you to ask questions, all your questions to the speakers. And you can do that at any time using the question and answer tool that you can find here at the bottom of your screen. And after the talks, we will go through the different questions, and the speaker, of course, will answer to them. One advantage of this Q&A feature compared to the chat, which is also available, is that you have also the possibility to like or to vote for the question that you find particularly interesting, and the question with the most like will be prioritized, and this will help the moderator. That's why we really ask you to use this Q&A for your questions and not the chat. Another thing I would like to mention is that this event was organized together with an amazing team. This is the team of Sense-Tribe, which is a consulting organization based in Spain. And today we have with us Laura Grassi and June Stefano. They will be coordinating everything in the background and making sure that everything works. And we also have the chance to have a third member of Sense with us, Marina Roa. She's extremely talented illustrator. She did, for example, all these images that we are using for this event. And she will be doing the visual harvesting, which means she will be drawing during the session and trying to capture the main message or the main aspects of the discussions. And of course, she will also share her drawings with us at different moments. And the last member of this hosting team is Marion Bettizo, my colleague from the Velux Stiftung. She is also with us today, and she will be helping particularly the moderator with the Q&A session. Another information for you, please note that this webinar is being recorded at the moment and that we are also live on YouTube. And of course, the video will be available on the Daylight Academy website in a few days. So we can start with the webinar. So I'm very happy to introduce the moderator of the day, Professor Debra Skene. Debra is a section lead of chronobiology at the University of Surrey in the UK, and she has over 25 years of research experience studying the human circadian system. So it's a real privilege to have her facilitating these exchanges today, and I don't want to wait any longer to give her the word. So Debra, please, the floor is all yours.
Thank you very much, Lydia. And a warm welcome to everybody in the Daylight Awareness Week. This is day 2 of this series. And today, the topic of our discussion is in the shade of the lockdown, how does the lack of daylight affect us? And I'm pleased to say that we've got 3 internationally recognized scientists to address this topic. I'll share my screen to introduce you to them. We've got the Professors Russell Foster, Sama Hattar, and Till Roenneberg, and essentially they will have 15 minutes, so a short talk, after which we'll have a few, if we have time, a few questions and answers. So please, you're encouraged to write questions in the Q&A. Also, if you like a question, signal that so we can get a sense of trying to address some of the more prominent questions in the series. The other thing is that we wanted to be very interactive. I mean, it's pretty crazy talking to over 200 people and not seeing any of you. So we just want to get a sense that you're there and you're ready to work with us today on this. And on that note, we're going to ask you to take your phone, please. Someone's not getting audio, okay? Can you hear me? Okay. And please now take your phone and go to www.menti.com. And when you get to that website, enter the code 4660291. And the first question we're asking you to write, please, is where you're joining us from, so we can get a sense of who is out there and where you are. Oh, someone from Finland, they were first, and Switzerland, and Brazil. And of course, the bigger the letters, the more people that we have coming from us. So of course, I should have said good morning to some people, the Brazilians, who's still pretty high in the top there, and I should be saying good evening to a lot of you. That's excellent. Could we have the next question, please? So the next, if you just press next question, what is the first word that comes to your mind when you think of lack of daylight? So we've got sleepy, winter, Brazil seems to be coming up, depression, fatigue, dark, sadness, happiness, cold. So lots of thoughts about lack of daylight. Indeed, disease, dullness, melatonin, circadian disruption, depression seems to be key. Thank you. And then finally, the next question, this is the last question. What do you hope to get out of this session? You just a small new information, enlightenment, nice, new information, knowledge, new ideas, learn a lot, overview, understanding, inspiration, difference between daylight and electric light. Good, well, thanks everyone. I know you're all out there. And so speakers, no pressure. They want new information and lots of knowledge. So on that note, it gives me great pleasure to introduce our first speaker, Professor Russell Foster. Russell is a recognized authority in studying the biological effects of light and its effect on the circadian system and sleep. And he's going to talk today about understanding how daylight regulates circadian rhythms. So, Russell, over to you.
Okay, can you hear me?
Yes, we can hear you now.
Ah, jolly good, fantastic. Right, I will then kick off and push the timer. It really is a great pleasure to join you all today, and thank the Daylight Academy and Lydia and her team, and for Debra for your introduction. Really what I want to do is provide an introduction to this whole theme today, and it's understanding how daylight regulates circadian rhythms. And part of this presentation will be indeed introduction, really to try and give you set the stage for my wonderful colleagues Sam Hattar and Till Roenneberg. But the second half of the discussion will be really on the impact of profound blindness on our sleep-wake and circadian rhythms. And as Lydia said, we take daylight for granted, and that's really where I'd like to start. We sit on a planet that revolves once every 24 hours. And of course, it generates the night and daylight. And this 24-hour oscillation is such a profound impact upon the ecology of the planet. Everything on Earth, almost everything on Earth, has evolved an evolutionary response to these changes. In us, we have the two states of sleep and consciousness. Sleep, we may have physical inactivity, but of course a huge amount is going on within the brain whilst we're asleep to actually allow us the conscious state. But the point I'm trying to make is that these are such very different states. They're underpinned by an extremely different physiology. So if we look at some aspects of our physiology, our biology, across the 24-hour day, and in these boxes here, each case you see approximately when we are asleep, and we see a 24-hour time base here from 2 p.m. to 2 p.m. And let's look at one rhythm. And this is melatonin, Debra's favorite hormone, which is produced
Russell, I believe your presentation is not on screen, so you would have to share your screen so we can see the presentation.
Okie dokie, I will start again. Okay, so could you hear me before?
Yes, we could hear you, and now we can
okay. You couldn't see me. All right, so I will skip.
Best if you put it in presentation mode so we see it even clearly, more clearly. Exactly, wonderful. Thank you.
Okay, so what I was saying is that we sit on a planet that revolves once every 24 hours. Profound differences in the ecology and our response to the day-night cycle has been to evolve the states of sleep, physical inactivity, but lots going on within the brain that define our ability to function during the conscious state. And where I got to was that these two very different states are underpinned by profoundly different aspects of physiology. And where I got to was talking about the hormone melatonin produced from the pineal gland. And what we see here in each of these boxes is a 24-hour time base, and the gray area is approximately when we sleep. And we see that melatonin rises in anticipation of the dark and the sleep state, and declines in anticipation of morning and the wake state. And this is melatonin, but we can take a whole number of different rhythms here. Growth hormone is released almost exclusively during the first part of the night. Core body temperature, a drop in core body temperature, probably important for the initiation of sleep. Cortisol, the stress hormone, rising in anticipation of activity. Changes in testosterone, and I can let you imagine what that may result in. And indeed, our ability to perform mental tasks such as multiplication activity changes across the 24-hour day. And the thing I want to emphasize is that these are under circadian control, which means that if you went into a deep dark cave, constant light, constant temperature, these rhythms would persist. Okay, so what do I mean by circadian control? At the heart of the biological clock that's ticking away within each of us is a paired structure down in the hypothalamus called the suprachiasmatic nuclei. And they sit either side of where the optic nerves enter the brain. It's about 50,000 individual cells. And what's important about these cells, that each one has its own clock. And that clock is essentially a molecular feedback loop, whereby you have a number of clock genes. They encode for the production of a protein. We have these clock proteins, which form a complex, enter the nucleus and actually inhibit their own production. And what this means that in each cell, you produce a cycle of clock protein production and degradation. And that is essentially the signal that is used to drive rhythmicity throughout the rest of the body. Now, we used to think that the SCN forced a pattern of activity on the rest of the body, and now we know that essentially every cell in the body has its own clock, and it's coordinated by the master clock within the SCN. But at the effector end, in all these structures here, regulating our physiology, this is sort of a local circadian control. Okay, so what we've shown here is an entrained circadian rhythm. And what I mean by that is that the peak of the rhythm occurs at the same time on subsequent days. But if we plunge this individual into complete darkness, we see that the rhythm persists. But in most humans, the rhythm is slightly longer than 24 hours. And so we see on each subsequent day, the rhythm gets longer and longer and longer. It fails to be entrained to the external world. So light is critical in setting the clock to the external world. If we think about what this looks like in terms of behavior, here we have again another 24-hour time base. Here's light, here's dark. And what I'm going to show you is roughly when we're asleep. And you see each line here is when we're asleep. But if we were plunged into darkness, then our sleep would get later and later and later each day. And we call this entrained, and we call this free running. And you'll see lots of examples of this in the rest of this talk and with my colleagues. Okay, so what happens if you're not entrained to daylight? What happens if the clock is just drifting through time and you're not getting enough daylight? Well, we can consider this in three domains. One is short-term disruption of the sleep-wake cycle through lack of light. And we can see, for example, loss of attention and high levels of microsleeps. About 100,000 crashes on the American freeway every year are associated with microsleeps, falling asleep at the wheel. You can increase the feelings of stress and anxiety, difficulty falling asleep, impulsivity, loss of empathy, failing to pick up those social signals in family and friends and work colleagues, and indeed a negative focus. You remember the negative things in your life, but not the positive things. Memory impairment, increased mistakes, clumsiness, reduce cognition, creativity, and information processing. And so this is short-term disruption of the circadian and sleep systems. Longer term, as you see in night shift workers, is associated with sustained physiological and psychosocial stress, increased stimulant and sedative use, immune suppression, which may underpin high rates of infection and cancer, increased cardiovascular disease, metabolic abnormalities such as diabetes 2. And if you are vulnerable to mental illness, then sleep-wake disruption as a result of not getting a strong daylight signal can increase mood instability, anxiety, paranoia, hallucinatory experiences, and indeed exacerbate the symptoms of bipolar and schizophrenia disorders. So these are really serious conditions that arise from disconnecting our internal timing systems from the external world. So how is the circadian system actually entrained by light? And for that, we look at the eye, and we can enlarge the eye and then look at the retina. And here we see the rods and cones, the classical visual cells of the eye, the inner retina, which do the first stages of visual processing before a signal is sent to the retinal ganglion cells, whose axons form the optic nerve. And we know there's a direct projection from the optic nerve to the suprachiasmatic nuclei. So light detected by the retina goes to the SCN and locks the clock onto the external world. What's extraordinary, however, is that you do not need your rods and cones to detect that light-dark cycle. No rods and cones, and this was shown in both animal models and in humans. So there's another receptor in the eye, and that receptor is based upon a small number of these ganglion cells. We call them photosensitive retinal ganglion cells, and they use a blue light sensitive photopigment that is called melanopsin. So you have a third receptor in the eye, which is primarily responsible for regulating the internal clock to the external world. But how much daylight do we need to regulate our circadian systems? We are exposed to a remarkably broad exposure to light. If we think of light intensity measured in lux, 0.1 lux would be something like moonlight. We go through a candle at 1 meter is 1 lux. Sunset can be as low as 50 lux. In the office, you'll be lucky to get 300 to 400 lux, and in domestic settings, such as our sitting rooms, you may go down to only 50 lux. Near a window, 3000 lux on a sunny day. Outdoors, in the shade, about 10,000 lux. And even in England, on a bright sunny day, you might get to about 100,000 lux. But the point is that it's our rod photoreceptors that give us our sense of black and white vision, detecting low levels of light. Our cones need more light to give us a sense of color vision, but there's plenty of light in the normal inside spaces for color vision. But for those photosensitive retinal ganglion cells, they need much more light relative to the visual rods and the cones. It's complicated because, first of all, we've talked about intensity. The intensity for detection by rods and cones is a fraction of a second, whereas these melanopsin-based ganglion cells require a relatively long duration of exposure, minutes to many minutes to hours of exposure. They also are sensitive to blue light, and that can influence their sensitivity, or the sensitivity of the circadian system to light. The time of day can influence how we respond to light. It's dawn versus dusk have very different effects. Whether we are agricultural workers and have spent a lot of time outside versus inside, and it seems that there's an adaptation to our light history. And also with age, it seems that in aged individuals, the whole of the circadian system, for reasons we don't fully understand, is less sensitive to light. Furthermore, if we look at our internal spaces, they really don't provide a robust enough amount of light for robust regulation of the internal clock. And in a sense, this can be made worse because of altered daylight exposure. Many of us get a period of daylight exposure, which can set our clocks to the external world. But If you are fairly old and you are in a nursing home of some sort, you may not see any natural light or very little natural light. Night shift workers have a very distorted exposure to the light-dark cycle. Office workers have a very different sort of exposure to agricultural workers. And if you suffer from neurodegenerative disease, there's a double whammy. It seems that mechanisms that are regulating the clock may be affected as a result of neurodegenerative disease, and you also tend to spend more time inside. In mental illness, again, there may be mechanisms as a result of mental illness that alter the sensitivity of the clock, but again, you're spending more time inside. And of course, the profoundly blind, which I'll move on to in a few seconds. The point is, in the aged community, all the way through these various categories, the whole situation has been made worse by COVID-19 and lockdown because there's been reduced opportunity to spend time outside. Now, in the profoundly blind, and what I mean by that are people with no eyes, for example, no matter how much natural light you see, it's not going to help you lock on to the external world. So let's just consider the profoundly blind for a few moments. What we can do is put a little wristwatch device to measure the rest activity cycle onto an individual. And the first person I want to look at is unemployed. And let me just move this. And what we see here is a 48-hour time base. Here's midnight. And you see that these deflections here represent when the individual is active. And you see subsequent days here. So this is midnight. They're going to bed after midnight. They're waking up variously, but it's a pretty stable rest sleep cycle, activity sleep cycle. This is an individual with no eyes. They lost their eyes as a result of a tragic accident. And you see that their rest activity cycle is drifting through time. And it really is profoundly difficult for these individuals. These are some quotes from individuals we have worked with. I'm at my wit's end, suffering from variable bedtimes and wake times. I'm slowly becoming socially isolated. I don't feel healthy anymore. I find myself getting irritable and not able to cope. So being profoundly blind detaches these individuals from the light-dark cycle and, of course, all the problems that result from a disrupted circadian sleep-wake cycle. What can we do for these individuals? And what we've been working on, and just this is the last few moments of the talk, are some perhaps ways in which we can fool the clock, its seen light, by using drugs. So we're using our knowledge of how daylight regulates the molecular clockwork to correct sleep and circadian rhythm disruption in the profoundly blind. So I want to declare some interests here. Part of the study I'm going to be talking about has been funded by venture capital through this spin out called Circadian Therapeutics, of which I'm a co-founder with Arti Jagannath and Sri Vasudevan. Okay, so this is the philosophy where a photosensitive ganglion cell detects light. It releases a neurotransmitter. It then triggers a signaling cascade. That signaling cascade then interacts with the molecular clockwork. The molecular clockwork then produces a signal which allows entrainment. What I mean by that is under a light-dark cycle, we go to bed and get up at about the same time. Under constant darkness or without that entraining signal, we drift. Give back an entraining signal, we can return once more. So we have full entrainment. Okay. So these are the mechanisms that we've unmasked, and I'll go through this quickly, and this is really for those individuals who want a few more details about these mechanisms. And what we found is that light will interact with one of the key clock genes, which is PER1 and PER2, which is part of the molecular clockwork, and interacts with regulatory elements in those genes. Basically, what happens is that glutamate and PACAP neurotransmitters are released from the photosensitive retinal ganglion cells. They increase calcium and cyclic AMP within one of the clock cells. That causes a protein complex to form, CRTC1, which binds to one of those regulatory elements, and we get the production of PER1 and 2. But it's more than that. There's another element which is activated by PHOS and its partner June, which interacts with an AP1 binding site. And this represents the second wave of PER1 and PER2 production, which acts as a signal, which, as I say, allows us to re-entrain to a light-dark cycle. But the point I want to make is that the effects of light on the clock are limited. And we know this because it takes about a day for every time zone we've crossed to recover. Why is that? Well, there's a second loop here. There's a protein called SIC1 that is also activated
Russell,
by light.
I'm calling you to...
Yep, 60 seconds.
Yes,
yep.
please.
Which then turns this whole system off. And you turn off the molecular signal which allows entrainment. So, my last slide. The logic of what we're doing is we're replacing those photosensitive retinal ganglion cells with a drug. And the two drugs we're working on are something that's going to mimic the effects of calcium and cyclic AMP, which is what light does exactly. But another drug is actually an antagonist to SIC1. And what that does is essentially make the clock much more sensitive to light. So what we're doing is fooling the clock that it's seen daylight. And so in our profoundly blind individuals, those individuals with no eyes, we're restoring a sense of time. So the points I wanted to cover is that daylight entrains the circadian system to the 24-hour day. Critically, this is achieved by specialized photoreceptors within the eye called photosensitive retinal ganglion cells. Lack of entrainment causes significant problems for health and well-being. Circadian entrainment requires relatively bright light of a long duration, so real daylight, and these cells need brighter light compared to visual responses. Eye loss and radical eye damage prevent circadian entrainment, but understanding the mechanisms how daylight regulates the clock is helping us develop drugs that fool the clock that it's seen daylight to restore a sense of biological time to the profoundly blind. Thanks very much for your attention.
Thank you indeed, Russell. Thank you very much. Of course, we have millions of questions, but I'll just ask one, because then we'll have to move on. Jens Christofferson asks that there are studies showing that higher daytime exposure to daylight ensures longer sleep at night. Wanted to know what your thoughts were on this, and what, in light of that, what should be a good recommendation?
Yeah, I think that a sort of robust exposure to the environmental light-dark cycle acts to stabilize the circadian system, of course, and that will stabilize the sleep-wake cycle. And so, you know, the classic experiment was done by Aus van Sameren on those individuals in a nursing home, where he increased the amount of light in the nursing home, stabilized sleep-wake, improved cognition in those individuals, and actually in their overall well-being. So light, as the questioner sort of indicates, is profoundly important in stabilizing sleep-wake and of course, overall health and well-being.
While you're listening to our next speaker, you're at liberty to answer some of the other questions that have come through. And then, of course, we'll also, don't worry, we'll also have some time at the end where we can pick up on other questions. So we'll try and get through everything. But at the moment, we're going to have a quick Zoom poll, I believe. And here is the Zoom poll for everyone. So you could just literally put a click. Are you an early bird? Are you neither an early bird nor a night owl? Or are you a night owl? So let's get a view from everybody of what they think they are, an early bird or a night owl, or neither one nor the other. It shouldn't take too long to get some details on how everybody voted there. So what does it tell us? Okay. Is that a lovely Gaussian distribution? Most neither early bird nor night owl, and then we've got quite a few more early birds than night owls, but a lovely, as we would expect, group of people. So thank you for that. And now it's my great pleasure to introduce our next speaker, Dr. Sama Hattar. Sama is chief of the section on light and circadian rhythms at the National Institute of Mental Health in Bethesda, the US. He's a recognized authority on the non-image forming photoreception, or as we call it, the non-visual effects of light. And he's going to speak to us today about light, circadian rhythms, and brain functions. Sama.
Thank you, Debra. I can't start my video for some reason. So if somebody can help me, it says unable to start video, but I will share my screen.
Okay.
All right.
Was working okay.
Yeah, but
There you go.
Okay, great. So I would like to thank the Daylight Foundation for inviting me to present my work on how light affects your circadian rhythm, sleep, and brain function. But before I start my talk, I would like to acknowledge the people who actually do the work and make me excited coming to work and work on these problems. It's really lucky to present after Russell Foster because you've already heard that light goes through your pupil to hit the back of your eye where the photoreceptors, the classical ones, rods and cones, are located. One thing I would like to mention is that light first experienced the ganglion cells, the one that actually connect the retina to the brain. So light has to cross all these layers before it hits the photosensitive parts of these extremely sensitive photoreceptor rods and the cone cells that allow you to see images. And you're possibly most aware of your vision when you look at images and you consciously see the environment around you. So for example, light received by a reflection from this image through the retina, through the optic nerve, and then projecting to brain areas, eventually to the cortex, allow you to consciously see this image. So although this image is two-dimensional, you could see here that there is waterfalls, there are cliffs and trees and even blue skies. But the eye detects light for functions that are incredibly simple, like for example, just controlling the amount of light that goes into your eye by regulating the size of the pupil. As you could see, if there is less light, the pupil is bigger than if there is more light. And as Russell beautifully showed us, circadian photoentrainment, which is simply adjusting the approximate day within organisms to an exact 24-hour period. So what is so nice about doing experiment in animals that this system is very similar between mice and humans. But what's really nice about mice is that you could genetically manipulate them. They don't have a variation in their behavior as much as we do. And you have a very strong behavior that you could measure. So just as Russell has showed you, if you put animals under 12 hours of light, 12 hours of darkness, mice are nocturnal animals, so the activity in the dark. But if you put them in constant dark conditions, you could see that they drift, as Russell beautifully showed you. It's free running rhythms. You could actually jet lag these animals. So if you bring them from New York to Amsterdam or Amsterdam to New York, it takes them 6 hours to adjust to each shift, just like us when we travel across different time zones. One thing that was really important to mention is that, maybe Russell was too humble to mention that, is that up to 1999, there were a lot of evidence that there may be other photoreceptors in the retina apart from rods and cones. But honestly, people thought that the individual who mentioned that are not really understanding the retina, because why would you have other photoreceptors that can complement such beautifully evolved photoreceptors to detect light? But it was really Russell Foster group in 1999 that did a very exciting experiment where they produced an animal that is completely image blind because they lack all the photoreceptors, rods and cones. Yet these animals beautifully photoentrained to the light pulse, similar to wild type animals. And to make a very long story short, it was really the pioneering work of Ignacio Provencio and David Burson, and then in collaboration with us when I was in King Wai Yao's lab, that we discovered that a subset of these ganglion cells, the one that see the light first, are intrinsically photosensitive. That means they respond to light independent of rod and cone input, and they project directly to the brain. So we call them intrinsically photosensitive retinal ganglion cells, or IPRGCs. Now notice that if you remove rods and cones, animals can photoentrain. If you remove melanopsin from IPRGCs, they become normal ganglion cells, but animals still photoentrain. Only when you remove rod, cone, and melanopsin, now animals will free run and be similar to the humans that Russell showed you that they don't have eyes at all. So these animals cannot entrain to the light-dark cycle, even though they still have a beautiful clock. So now we know, and we only knew this in 2002, believe it or not, that the retina doesn't have only 2 types of photoreceptors, but actually 3. You have the classical photoreceptors, rods and cones, but also these atypical ganglion cell photoreceptors that are themselves photosensitive through melanopsin. But if you remove melanopsin, rod and cone can still signal to the brain because they act as retinal ganglion cells. Because they project directly to the brain, we were able to actually trace these cells from the retina to the brain. And you don't have to know the details. We just were able to label them with a blue color and label their axons, which project to the brain. And this is the moment that doing science pays off, and where you feel that the whole world cannot, fit you because you're so happy. When I got the brain from these animals, what we saw was really remarkable. Here's a magnification of this area here. You could see that these cells project directly to the suprachiasmatic nucleus. And if you've heard Russell talk, this is the area where the circadian pacemaker is located. But what was really remarkable to me, and quickly was the interest of my lab later on, is that these cells don't only project to the circadian pacemaker, but they project to many different brain regions. You don't have to know the name of them, but you have to know that they have incredibly important functions. Some that are important for pupil constriction, some that are important for your sleep and alertness, and some even that are important for your mood regulation. And if you remember the first poll that Debra told you to take, depression was one of the biggest problems that people felt with the lack of light. And this may not be involving the suprachiasmatic nucleus, as I'm going to show you in a bit. For the aficionado, for the people who are more experienced in this field, I would like to just mention that now we know that even these IPRGCs are incredibly complex. I'm happy to talk more to you about that and to show you the data. And that if you remove the most important one for non-image, the M1 population, now you produce an animal opposite to the one that Russell had showed you before. Now you produce an animal that is able to see complex image formation, but is not able to detect the simple absence or presence of light. This to me still is very surprising because it's kind of complicated to think about. This is similar to a great mathematician where she can solve the biggest problem in math, but she cannot tell you what 1 + 1 equals to. So that's quite remarkable to me. So this told us, though, that retinal cells that influence circadian photoentrainment are distinct from those that affect image formation. So honestly, up to recently, and still a lot of the people in the field think this way. I mean, for example, this is a review from an amazing scientist in the field, Mick Hastings, that I have complete respect for. And people think of light and dark affecting the suprachiasmatic nucleus. And as Russell showed you beautifully, the SCN coordinate all these beautiful oscillators in your body. But I'm a more daylight person than clock person, to be honest. So I would like to tell you that actually there is something more interesting than that. And I'll tell you why it's more interesting, because now we know that these IPRGCs not only project to the SCN, but project to many different brain regions that project to area that affect reward, like the ventral tegmental area, that project mood, like the raphe nucleus, that project even executive memory and reward, like the ventral medial prefrontal cortex and the nucleus accumbens. So there is a lot to light that can influence you as a person or as an animal that is not directed through the circadian SCN. So then, because we saw this incredible diversity of IPRGCs projecting in the brain, we wondered whether this dogma in the field that all these disruptive light environment can cause mood and learning difficulty through circadian and sleep problem is completely the truth. So what we wondered ourselves, whether light can directly affect mood and learning independent of circadian and sleep problems. And I'm really summarizing tons of research by a lot of my people in one slide, but I would tell you that we were stunned to find that although this circuit may be correct, and we're not going to say if it's correct or incorrect here, but I'll be happy to answer a question later, that there's clearly another parallel circuit where light through IPRGCs can directly affect mood and learning and memory, independent of disruptions of circadian rhythms and sleep. And what was really remarkable for me is that conscious image formation, at least in rodents, do not play a role in these effects. So we don't know if this is the case for humans, but what we know that in rodents, you need this IPRGCs to affect mood and learning by light. And we recently published an extension of this work, and it got reviewed by Yanar and Halasa in Cell, where we found that these IPRGCs that affect mood project to a previously unknown region in the brain known as, we now call it the perihabenular nucleus. It's part of the thalamus, the dorsal thalamus, where ambient information can somehow affect this region to project to regions downstream that affect mood and even cognition and memory. One thing that is really exciting about this region is that it is located in an area in the brain that seems to be specializing in stress responses and depression and reward. So close to this perihabenular region, you have the lateral habenula, a very well-known region that is important for depression and reward, and the PVT, which is also important for stress responses. So it's really exciting that light effect in depression is clustering with all these stress-related and depression-related areas in the brain. Remarkably, although the PHB does project to the medial prefrontal cortex, which is important for executive memory, the effect of light on memory seems to require the SCN, but in a clock-independent manner, which is really remarkable because it seems that light through the SCN somehow lead to effect on the hippocampus. And I'm sorry, I'm going to go back to one slide. You could see the hippocampus is actually very close to this region, but the hippocampus does not receive direct light input from the retina. So now we know that in addition to circadian photoentrainment, which is absolutely important and critical for both us and animals, there are a lot of other pathways that don't seem to require the SCN, but are important for the effects on our physiology and behavior, especially on our mood and learning and cognition. And one thing that I like to add that is really interesting, that even the SCN may not be as simple as we think of it as just simply a circadian pacemaker, because the SCN can act as a pacemaker through photoentrainment, but it could also act as a simple conduit for light to affect our cognition and learning and memory. And we recently, in collaboration with Tiffany Schmidt, who has her own lab right now, published a very exciting paper in eLife, where we showed that even for sleep and body temperature regulation, there are areas outside the SCN that are important for their acute effect of light on these behaviors. So these behaviors can be regulated by the SCN to be an exact 24-hour period to entrain. But if you get disruptive light at night, you're most probably engaging areas that are not the SCN to directly affect sleep and body temperature. And now we have a study to try to pin down where these regions are and how they control sleep and body temperature. And so that's the question that my lab is currently trying to answer, to figure out which are the brain regions that can acutely affect sleep and body temperature independent of the SCN effect. Finally, in a very recent publication in Nature, we have found something even more remarkable, and I'll be happy to talk about this more in the question and answer session. We found that developmentally, IPRGC innervation of the suprachiasmatic nucleus is important for a really exciting function about anticipation of food. So if humans are provided food at a certain time of the day, they start to anticipate this food even before you give it to them. And remarkably, this food anticipation does not require the SCN, but requires some areas in the brain that we still do not know, and another area called the intergenital leaflet that I'll be happy to talk about, to actually set this system up. So what we're now starting to find, a general mechanism of light and the circadian clock and other function, is that the circadian clock is important for entraining everything to a day-night cycle, but that there are other effects of light that can manage acute effect of changes in the environment. So for example, let's take food, because this is something that I'm really excited about right now. If you are hungry, it's actually the arcuate nucleus that determines the hunger level. So hunger is mostly determined by the arcuate nucleus. When you're gonna eat on a regular cycle is mostly determined by the SCN. But if you have changes in the availability of food, or the food start appearing at the different times of the day, is actually a completely different system that requires completely different set of brain systems to allow you to anticipate the availability of that food.
Can we conclude, please, Sama? Yes,
Great.
perfect.
So I would like to thank the previous member that I didn't thank before, that really did all these experiments that I showed you, the foundation that funded and the NIH that funded this work in the extramural, and the mouse TriLab, Lorenzo, who's still at Johns Hopkins, and the intramural research, and Justin, who's still at Hopkins, who also helped me do all these experiments. Thank you very much for your attention.
Thank you. Thank you very much indeed. And yes, we have some questions. I would encourage the audience to keep them coming, but also please, if you do like somebody else's question, and this gives us a chance to look at what is the most popular. I'm trying to see whether there's anything specific that might relate to your talk, Sama, something quick and easy that we could do. There's some things I've just, obviously they sitting here at the bottom here. There've been a lot of questions though about intensity and about light from things at night, and really the threshold that IPRGCs may have. So for example, if you're exposed to daylight inside an apartment near a window on a sunny day, is that going to be enough to activate the IPRGCs?
So this is really one of the most important questions we have in this field right now. And the amount of information we have, especially in humans, and even actually in other animals, is nearly zero. So what we know for a fact is that different behaviors require different intensity of light. So for example, the circadian photoentrainment is very sensitive to light. But the effects of light on mood and learning and memory require a lot of light stimulation. In addition, Tiffany Schmidt has beautifully showed that IPRGCs themselves are variable. Some IPRGCs are incredibly sensitive to light. They are breaking the dogma that they are not sensitive. And this is not due to the rod and cone input. This is due to their intrinsic responses to light. In fact, Tiffany had showed that some of them are as sensitive as nearly as cones. So this is something that we still don't understand. So it's very important, if we want to proceed further and understand how light is going to affect us in general, is to try to understand what is the sensitivity of IPRGCs in a human for different functions. So we need much more research on humans that measures what is the sensitivity for the pupil constriction, what is the sensitivity for melatonin suppression, what is the sensitivity for alertness, what is sensitivity for circadian photoentrainment. So that's a very important question that needs to be studied further.
Thank you very much. And there's a question regarding the different subtypes of receptors that I think you'll be able to answer offline when we go on to the next session. So thank you, Sama, and I'll see you at the panel discussion later on. So we now have another Zoom poll, please. Right, so here we have that the EU may abolish seasonal clock changes. In your opinion, which time should be kept all year round? So you've got three options: daylight saving time, also known as summertime, or standard time, also known as winter time, or you really don't care. So if you have an opinion about that, if you could just fill in one of those options, and we can get a sense of our audience and what they would be supporting if the EU were to do this. And what does it look like? Okay, so we must have some chronobiologists in the house because standard time is winning, and daylight saving time 36% compared to winter time at 48%, and people don't really care, 16%. So thank you for completing that. And it comes at a perfect time because our final speaker is Professor Till Roenneberg from the LMU University in Munich. And Till is really a pioneer in our field of chronobiology. One of the big recent contributions he's made to the field is his Munich chronotype questionnaire, and how that can assess chronotype based on sleep timing. He's also introduced us to the concept of social jet lag. He has opinions about daylight saving, and he's also going to present something in the shade of lockdown and speak specifically to some data that he's recently collected. So without further ado, I'll hand over to Till. Oh, I have one other, I've made one other small omission. I forgot to mention, and that I could do it at the end. Till, you're on.
No,
I made a mistake.
Do it now. Do it now.
No, it's fine. We'll do it at the end, and we'll have more time for it. So it's fine.
Nobody has time at any end. Okay, thank you very much. I'm going to bring what Russell and Summer has told you about the mechanisms and the incredible biology behind the circadian clock, behind mood, behind light, and so forth. I'm trying to bring it back into everyday life. And I will go through three chapters here, short chapters, of course, and that's the circadian clock, light, and chronotypes. I don't have to go into the circadian clock. I don't have to go into light because both Russell and Summer have done that perfectly. But the variability of this biology still has to be stressed, and I'm going to go into it. And then I'm going to I'll tell you about real world life, big real world experiments, because I think that we can do experiments without having to set up a laboratory in the real life with humans as the subjects. And then I will come to the theme of the afternoon or evening in the shade of lockdown, and we've done an experiment, a real life experiment towards this end. So the question of chronotype, which you answered already, was that there are fewer very late types, there are fewer very early types, and there are many in between. So a neither type is something that has been called neither type. I really don't like that, because if you ask people how about their body height, you may have very short people and very tall people, but you wouldn't call the majority neither too short nor too tall. So I think it's a genuine distribution of chronotypes. It's a value that goes from very early to very late, with the majority in between. The reasons, the very early types are called larks, and the very late types are called owls. And the intermediate types have never had a name. I want to call them doves to give them a bird name too. So the reason why they are different has three backgrounds. One is the genetic disposition. which you've already heard about, and there's a variance and the variability in the genes that are responsible for not only the clock itself, but also for the light input pathways. Then, interestingly, there is age. We are quite early as children. We get later and later in adolescence, and then we get early again from the age of 20 on until we reach what Germans call the senile bed flight, where we wake up very early despite of what we want to wake up at, and can't fall asleep anymore. But the most important ingredient that determines whether you are early or late lies in the daylight exposure. And I say daylight exposure not only because this is the Daylight Academy, but because daylight is so much more powerful than any other light that we are exposed to. So these are very important in making in making different chronotypes. There's something else that has come with industrialization. Now, pre-industrialization, we were true day active animals, running around during the day and sleeping at night, and there was no real structure in our sleep from day to day. There was variation, of course, but there was no real structure. Now we have changed that by being inside most of the day, and thereby reducing enormously the amount of daylight that we are receiving. And we have increased the light at night, so therefore decreasing the historical darkness that we were exposed to, and thereby we have completely flattened the Zeitgeber strengths that we expose our clocks to. And this has led to the fact that, in combination with the times that society wants us to work at, first of all, under these conditions, most clocks become later, and therefore we cannot start to sleep as early as we would have, or we did when we still were camping or living without electricity. Now we can fall asleep very late, but we still have to get up late. We accumulate during the work week, we accumulate sleep debt, and therefore we sleep in, if we can, on weekdays. The thing that we have created since the pre-industrial times is we used to have, in those days where we only had fire or the sun as light, we used to have a light-dark cycle, and the light-dark cycle entrained our circadian system, and the circadian system entrained or controlled, amongst other physiological things, that when we slept. Over the time now, we have almost constant dim light, which we expose ourselves to, and the only time when we expose ourselves to darkness is when we sleep, which is basically not only an input of the clock, but also an output of the clock, and therefore we created a short circuit. Now, when you look at sleep logs or any other measurements, you will find this remarkable structure, weekly structure of sleep. Namely, you get up at a certain point during the week, when most of the times the alarm call pulls you out of sleep, and then you get too little sleep, and then you have to sleep in order to catch up on the weekends. Now, this difference in when we sleep on work days and when we sleep on work-free days, we have called social jet lag, because it reminds us of people who fly to New York on Friday evening and come back on a Monday morning. And this is my icon for social jet lag, because social jet lag needs an alarm clock, and only 13% amongst the population does not suffer from any kind of social jet lag. So social jet lag is when you live biologically in a different time zone than the work time zone you want to live in. It has enormous consequences, and this slide shows something quite boring, because it's well known that we need more sleep when we're young. This is age down here and sleep duration here. We need less sleep when we get older, but we knew that. What we didn't know is that if we separate the two in how much sleep we get on free days and how much sleep we get on work or school days, you can see the huge discrepancy during adolescence and puberty between what we need, what we try to get back on free days, and that what we get during the school or the work week. Now, this is incredibly interesting and important because the more social jet lag you suffer, the shorter is your sleep duration, and the higher is your possibility of having social jet lag. In the shade of lockdown, big real world experiments, they are out there, and we've already had a poll concerning these outcomes. And one of the great things about large databases is that you can ask questions across the world. And here's something that is quite systematic, and that if you go within a time zone, and this is the Central European time zone, you find that in the East, people are very early, and light green is very early, black is intermediate, and red is very late. So you find this gradient within the time zone from east to west, actually it's from southeast to northwest. And you find that these poor people in Galicia still have to live on the same time as the people in Prague, and they are of course much, much later. So we are still hamsters, we are still bean plants or any other organism. Our circadian clocks go by the sunlight. This is another wonderful story by Jontella and Mazzona, who show how within time zones in the US, when people switch off their lights, and this is an entire time zone where you have the same social time for everybody, but people in the east of the time zone switch their lights off earlier than in the west. So the biology is consistent with our knowledge of what we're doing and what the clock is doing. And the consequences are enormous, because if you look at in the time zones from east to west at health, the health goes, I'm not going to go into the details, these things are all published. The health goes down from east to west, and then at the time zone border, it jumps up again to a better health quotient. Here is a big world experiment, which shows the standard time, which the majority of you have voted for, and the standard time within the Central European time zone, which is green here, and England and Portugal, GMT it's yellow here. Iceland is also in that time zone, and this is the Eastern time zone in blue. So the green time zone should only actually be in this green background, because that's what the intention of time zones originally was. All these France and Spain should already be in the yellow time zone. Now, if you go to DST, the biological time zones or the sun time zones are even worse, because now we all here in Central Europe live according to this time zone here, which is east of us, which is intolerable. So what we suggest as chronobiologists is that we not only abandon this crazy situation, which is increasing health risks, but that we actually go back to the original ideas of the time zones. And to finish up, I've come down to what we've done in a large survey in conjunction with the lockdown situation, the pandemic, and these are the questionnaire, the study cornerstones, how many people we have, languages. We've done it in many populations and in many languages. In many countries, the questionnaire was online from April 4th to May 16th. And the main issue that we found was that outdoor daylight exposure was cut into half during lockdown, which has an impact on our chronotype and our phase of entrainment, as you've heard from the two other speakers. Now, if you look at the general data of the over 7,000 people, there are remarkable determinants almost in the data. What you see here is a correlation or an XY plot between what the people's chronotype was before the lockdown and what it was in the lockdown. And you can see that there is not much change, but there is a general shift towards a later chronotype, which is consistent with getting less light during the lockdown. If you look at sleep duration, you already find what we have called a rubber band effect. Those people who get far too little sleep during the pre-lockdown era increase their sleep most. And what you get is, as a determinant, if you make a continuous moving linear regression here, is that about 8.5 hours is the duration of the average duration of sleep where people don't change their behavior from pre-lockdown to lockdown. So that's a very, very good hint towards what we need as a population on average. We also see that 20 minutes of social jet lag seems to be tolerable, because you can see that anybody who has more social jet lag is responding to it quite vigorously, whereas 20 minutes doesn't lead to any behavioral change between pre-lockdown and or pre-social restrictions and in social restrictions. So 20 minutes of social jet lag seems to be tolerable. That is very low, and most of us get more than that. If you go into the ages, and I want to cut this very short, this is becoming even more interesting. Chronotype is still quite parallel to a one-to-one relationship in most ages, except for the very young ones, which are suffering most and therefore correct most of their biological behavior during the times when they don't have to use an alarm clock. This is sleep duration, and you can see how wonderful the non-changing numbers. So these red points are when the population under this age group does not change their behavior. If they sleep shorter pre-social restrictions, they change their behavior. And the younger they are, the more sleep they need. Social jet lag, again, is across all ages approximately, except for the few people who don't suffer from social jet lag very much because they're already retired. But most people tolerate something like 20 minutes. With that, I would like to thank all the people who are involved in all of these big studies. And thank you very much for listening and being part of this fun webinar. And I give it back to Debra to do whatever she needs to do now.
Thank you very much indeed, Till. And thank you for keeping to time, which gives us more time for questions. And I was struck by one here from Sarah Rees. Where is it? It's to do with social jet lag and babies, because as you know, people who have babies are their effective alarm clocks, very effective alarm clocks.
Yes.
So her reasoning here, so if this counts as a form of social jet lag, perhaps it's part of the evolutionary history, our evolutionary history, because humans breed. And so it would be surprising if we don't have any adaptations to ameliorate this effect, because this would be something.
I see that point, and it's a very good point, but I slightly disagree, because what babies basically do is keep mothers awake, especially mothers, because at the early stages because of breastfeeding. So that's nothing to do with timing of sleep. That is something to do with constant interruption of sleep. The fact that babies are early or much earlier than adults is more recent, because if we take the population camping, most of the population will actually wake up very early and therefore the early wake up of small children or babies does not create any social jet lag. It is a recent phenomenon that the adults' clocks are so late compared to the baby clocks that the irritation is not only across the night by waking up parents or mothers especially, but by also waking up early. And this is going down because the more rural, the more urban children, even young children get, the later they will wake up. So I disagree that babies are social jet lag, because social jet lag is when you wake up too early, when you are woken up too early according to your body clock, and not just being woken throughout the night.
Okay, thank you for that clarification, Till. What I'd like to do now is correct an error earlier. I should have mentioned that we have Marina, who...
I can't hear now.
Can you hear me?
Now,
yes.
Now it's back.
Yes. Marina has been drawing away during our webinar and doing cartoons about the subject. So I'd like to invite her to show us some of the things, before we start our Q&A, some of the things that she's been able to do during the session. Have we got her screen? Or there's Marina. Thank you.
Thank you, Debra. Yes, I'm going to show my screen for a second.
We can hear you. So
that's great. Let me start from the beginning, and I will share my screen. Okay. Whoops.
Did you do that drawing too?
Sorry?
Did you do that drawing too?
Yes, I did that drawing.
That's
yes.
a very nice one.
Oh, thank you so much. Yeah, I just did a few, capture a few ideas of each of the talks. This was from the first talk.
Where's Russell? Has he already gone back to the Queen?
I don't know.
No, I'm here, listening calmly, carefully.
We can't see you.
Oh, I'm typing away to try and answer some of the questions.
Oh, super.
Oh, good for you.
This image was from the second talk. You were explaining a lot of information related with receptors. There are more receptors than we thought, and how they affect to the brain, different areas, even for beyond the brain.
Wow, impressive. Nice.
Fantastic.
And I was
so
trying
quickly.
I was trying to do a little comic about explaining social jet lag at the end. So you stay awake on Monday night, and Tuesday morning you go to work, and then Saturday, Sunday morning you sleep as much as you can. So thank you so much for your attention.
Yes, that's lovely. Thank you very much indeed.
You're very welcome.
We might want to borrow some of those for our slides.
Yes, absolutely.
Oh yeah, definitely.
Good, thank you. So now we do have time to have some Q&A, and we've got all the questions here. We're trying to, of course, be fair to everybody, and I'll try and find some questions that I think all the panel might have an opinion on. One of them that's coming through quite a lot is, is there a difference, the biological effects of artificial lighting or indoor lighting compared to natural daylight?
So I could start first, if you don't mind.
Good, thanks, Emma.
So I think the figure that Russell showed beautifully about the different intensity of light exposure across different environments just tells it all, because yes, you could cause some effect with dimmer light, with the 300 lux that you get if you're lucky in the office environment. But as I told you, there are so many other effects of light that have higher, they need more amount of light to affect than circadian photoentrainment. So if you want to get the full aspect of the physiology, I think, of the benefit of light, I think the daylight will be much better. And this is, again, this is hand waving, but would be better than artificial light.
Yeah, I would add, I think it's also fascinating that many researchers, none of the ones here, hold the mutually exclusive idea that the circadian system requires bright light to entrain, and yet light from screens, which is extremely dim, will also shift the clock. But actually the empirical evidence for that is very, very slight. I mean, the classic study that Chuck Seisler did, showing that 4 hours of screen use on the brightest intensity of the screen for 5 consecutive nights delayed sleep onset by 10 minutes. So, and I think Mary Carskadden said, well, it might be statistically significant, but it's biologically meaningless. And so I think, we've got to be slightly careful here, because as Sama has said, whilst the circadian system may not be sensitive to that kind of low-level light, the alerting systems may be. And so I think we need to disentangle which elements of light are interacting with which elements within the brain that are being regulated.
Yes, I think that's the right point. I think we still don't know enough about the different effects of light and the different thresholds in terms of intensity, but probably also in terms of wavelength and in terms of timing.
And duration, of course, is absolutely critical. So I think that it's very difficult for architects, because they're looking to us for answers. What is the sort of light that will be optimal for the circadian system? And we can't provide the answer at this stage, because it's going to be an integration of intensity, wavelength, duration, light history. And we need field studies. We've got to get out there, measure light in the real world, and correlate that with entrainment, alertness, subjective mood, and all the other things. It's doable because of the advanced wearables these days, but we don't have the data yet.
Debra, can I just quickly, there is a question that is always arises about nocturnal and diurnal, if you don't mind, because it's
yes, please.
Because a lot of people think nocturnal animal by definition hate the light, but actually I would like to mention that this is completely not true, because if you keep rodents in constant darkness, it's actually a big factor for inducing anxiety and depression-like behavior in rodents. So even animals that spend most of their time in the dark, they actually still benefit from light, as Russell and Till has said, for entraining and strengthening the circadian system, among other things. So just because something is nocturnal and diurnal doesn't mean that the circuits are different first, and doesn't mean that they will react differently to every aspect of light. So it's just where they choose their activity to be active or inactive.
Yeah, it's remarkable. If you hold a mouse under a dim light-dark cycle, they actually run in the light and sleep in the dark. They become diurnal if the light is dim enough, so they can actually
if you look at them in the wild, that can also happen.
Yeah, absolutely.
Noga has done beautiful experiment on these rodents coming in the morning from their burrows and just bask in the early sunlight, maybe just to adjust their clock or for warmth or whatever, but clearly that they are not just sitting underground all the time in complete darkness, that they go out close to their burrows, but they are exposed to incredibly bright sunlight.
Sorry to interrupt you, just Russell, could you activate your video? It would be nice to see you too, with the other speakers.
Okay.
Oh,
There you go. Yeah.
he's still there.
I just thought it would be easier on the eye if you didn't see.
Yes, that is true.
While you're there, Russell, there's been quite a bit of a few questions regarding if you are going to use drugs to, fool the clock.
Yeah.
How damaging? What other side effects, non-clock side effects, might we expect?
This is a critical thing. I mean, we've got to the preclinical stage where we've tested one particular class of drugs, for example, very rigorously in mice. And actually, it's a repurposed drug. So what we've done is screen a large number of drugs for their impact upon the clock. And 5 classes came up which hadn't been identified before, which are very effective at shifting the clock. One of these classes of drugs was actually developed to try and cure Parkinson's disease, went to advanced clinical trials, had no side effects, but it didn't cure Parkinson's disease. So for this particular one, we can move very quickly from the mouse to the human trials, which hopefully will start. They've been delayed because of COVID, which will start, and we've even got dosing measures for efficacy and toxicity. So it's an incredibly good question, and we are exquisitely sensitive to those particular problems.
Thank you. And now we've lost Till. Has he gone have you gone to visit the Queen, Till?
No, I have not. Although the Queen always says, if you see one, use it. No, I've just put on something on the stove.
There are quite a few questions coming up for you as well. There's one about what were your thoughts from Ellen Stowart? What are your thoughts on the variability or lack of between the age groups in their sleep need proxy in your new data set?
I don't understand the question, because the new data set sort of supports what we were knowing all along. We knew that from the graph that I've also shown, how much sleep young people use or take on weekends, that they needed to sleep longer. And since they were getting almost as little or even less sleep than normal adults, or consolidated adults, they needed even more sleep. So there was no surprise in the new data. What was surprising in the new data was the accuracy by which you could pin down at what average sleep need people did not change their behavior, and that was 8.5 hours. In which, looking at the data very carefully, which we didn't have the time to do here, what you see is that we probably have overestimated how much of the longer sleep on weekends is due to catching up with sleep. We now believe that most of it, 80% of the oversleep that we do on weekends, is what we really need and not what we use to catch up. So these new numbers are incredibly important for understanding how much sleep we need, because we never know how much sleep we need, since we don't have a measurement of sleep quality that can tell us if we slept enough. But these new numbers are telling us more and more towards the fact that we actually do need more sleep than we thought. And experiments in the lab have actually, where you forced people to sleep much longer than they were used to, have shown that the sleep need was larger than we ever thought.
Thank you. And probably one final question that was addressed to you, Sam, but perhaps Russell has some opinions too. What about extra-retinal photoreception? Your views?
Well, of course, I did my PhD on extraretinal photoreception.
Yeah, but Sam is allowed to ask for an answer.
Well, I think there's an interesting paper that's just emerged suggesting that OPN5, another photopigment molecule, could provide a light input for the regulation. I think it was respiration or heart rate. I can't remember the details.
Body temperature.
It was, oh, sorry, body temperature. Yeah, I think there's some other stuff that hasn't been published on heart rate. And so one would never, ever exclude, you can't prove a negative, but on the basis of the fact that if you don't have eyes, you can't entrain, I would say that there's no extra retinal input for the circadian system, but there may be other systems that require a light input. an extra retinal light input.
I would
Sama,
say,
last word for you.
I would say that there's still, apart from IPRGCs and rods and cones, there is not a single cell in the body that can respond to light intrinsically. So how do they do it? They clearly somehow are involving light, but how do they do it? As Russell has said, we still don't know. Are there extra photoreceptors? Even the pupil muscle actually is highly insensitive, and we don't know if it's even light responsive. So they require amount of light that literally you have to look at the noon sunlight directly to activate these cells.
But Sama, surely my skin cells are light sensitive.
Absolutely, they are light sensitive by mobilizing the color, but I'm talking about responding in a physiological, electrophysiological, like a photoreceptor. So you're absolutely right. I would say, yeah, we still have to see that responsive. We still have, just like what Russell suffered for a year, till you show it, till Eggy and David Burson showed it. It's hard to say there is something.
Yeah, I mean, I think it's very interesting to ask you a question. Why have the mammals across the vertebrates exclusively lost extraretinal photoreceptors when the birds, the reptiles, the amphibians, the fish have pineal, parapineal, hypothalamic, and other photoreceptors all over the central nervous system? And the partial explanation, or the explanation that we all sign up to, is that because mammalian lineage is unique in the fact that it's gone through a nocturnal bottleneck. So when the archosaurs, the ruling reptiles, dominated the daylight environment, our ancestors were emerging from their burrows, and there simply wasn't enough light at that time for long enough to support a photoreceptor that was embedded somewhere within the brain or under the skull or some other non-retinal site. So we retained our photosensitive ganglion cells, but we lost everything else.
Good. Well, of course, we could go on for hours, as we know, and some old familiar stories, as well as challenging new questions that are arising in some fantastic new research. So thank you all, the speakers, for speaking today, and all the participants that have joined us. It's been a pleasure to be amongst you. We've got one last Mentimeter quiz. So if you wouldn't mind going to your phones for the last time, and that's www.menti.com, and use the code 8128427, because we'd like to know what you're going to do now, thinking of activities, light seeking or non-light seeking. So will you go outside and fill up with daylight, if you still have any day where you are, or will you just shut down your light from your computer? Will you tell your friends to watch the recorded version? They need to know. Just clear your head with Netflix, or sign up for tomorrow's webinar. So looks like we've got sort of equal choices all round as we go along. But I think it's been super that you've all interacted so well. And I'd like to thank everybody, and finally to hand over to Lydia, who will close the meeting. Lydia.
Yes, thank you, Debra, and thank you everybody also for this last great answers. So now I know that you all have nice plans for after the webinar, so I will keep it short. Thank you very much to everybody again for the participation, for all the interesting questions, and of course a big thank you to the speakers and to the moderator for this really great webinar. And just a last word, and I will share my screen again. The Daylight Awareness Week is not over yet. We have a last session tomorrow. And of course, you are welcome to join us again. It will be a roundtable with 6 excellent speakers representing very different disciplines, ranging from architecture and art to ecology and chronobiology. And together with this very diverse panel of experts, we will explore how all this scientific knowledge that we have about the benefits of daylight, including what we heard today, how this knowledge can be applied to make our cities and our built environments healthy and sustainable. So also a very interesting question. So if you haven't registered yet, feel free to do it. You just have to go to the Daylight Academy website and you will find all information there. And if you have questions, of course, don't hesitate to contact us. So let's close the session here. Thank you very much again and have a nice evening.
Bye-bye.
Bye.
Thanks. Bye-bye.